this session. Today is the fifth day and sixth lecture of this session. On that day, the eminent resource person, Professor Dr. Amal Kumar Mondal, is present, and Sir discussed on the topic chemotaxonomy, its application in plant systematics with special emphasis on Sementides. So, I would like to request Mr. Arjun Patra to introduce Sir in front of participant. Please, over to you, Arjun.
Hello.
Yes.
Am I audible?
Okay. Yes, yes.
Good morning to all of you. First of all, I would like to welcome Professor Dr. Amal Kumar Mondal, and it's an opportunity to me to introduce such eminent speaker in front of our participants. Professor Amal Kumar Mondal presently engaged with Department of Botany and Forestry, Vidyasagar University. He has completed his MSc from University of Burdwan and PhD from Vishwabharati University, Santiniketan. His specialization on taxonomy as well as his broad area of interest is molecular taxonomy, ethnobotany, chemotaxonomy, paleochemistry of the taxonomy, as well as different medicinal plant studies. Sorry for inconvenience. Professor Mondal has published so many journals, articles, about more than 130 in international publication and more than 84 in national reputed journal. He has published book as well as edited book more than 25 in international and national reputed publishers. More than 10 students have been awarded PhD under his guidance, and presently more than 10 students working under his guidance. He has also a fellow of Linnean Society from London, as well as fellow of Indian Association for Angiosperm Taxonomy. He has awarded in Bharat Award from IIFS in 2013, and also awarded Adarsh Vidya Saraswati Award from GMC Ahmedabad in 2018. He has also awarded Distinguished Faculty Award from Venus International Friendship Society in 2018. So Professor Mondal completed many projects from UGC, DST, Boost, Government of India as well as Government of West Bengal. Presently, he is the coordinator of UGC DRS SAP2 and DBT Boost project in Department of Botany and Forestry, Vidyasagar University. He has completed many years in as a head of the department in Botany and Forestry. So now I requested to Professor Mondal to deliver a lecture on chemotaxonomy. I think this topic will enlighten and enhance our knowledge as well as the knowledge of students, academician, research scholar, and also faculties from uh, different college, different university in around India or abroad India. Thank you. So, sir, please start your lecture. Sir, over to you.
In P.K. College, and one of my other student, that is Obik Samanto, also established in that is P.K. College, and my Mumtaj Khatun, established in SSB College. So thank you very much for giving me this opportunity to share my knowledge, my research on the relevant topic, which is included partly in the honors syllabus and in the master degree syllabus. So thank you all, and just I am trying my level best to enlighten this interesting topic, chemotaxonomy or chemical taxonomy, its application in systematics with the special emphasis on systematics. Plant classification basically based on morphological characters. We know that the first this type of classification identification started by Professor Linnaeus. Carolus Linnaeus is the father of taxonomy, totally based on morphological characters. And he proposed classification, that is artificial system of classification. And after that, there are numerous taxonomists worked under different types of approach. Second, that is Bentham and Hooker. We can call this, their classification is natural system of classification. And after that, phylogenetic system of classification proposed by Engler and Prenter, Hutchinson, John Ray, etc. And lastly, with the development of molecular, chemical, and other ultrastructural aspects, the latest classification based on molecular characters. And we already know that APG1, APG2, APG3, and APG4 now. And APG4 last classification published in 2016, fully based on molecular characters. So since from Linnaeus to APG, it's a tremendous work by the taxonomist, systematist, molecular biologist for this enhanced development of taxonomy and systematics. APG system is difficult. It's not application in the practical classes, but it has tremendous positive characters, that is based on only molecular character. And we know that molecule is the final destination of all the biologists on the evolutionaries, on the evolution point of view. First of all, Linnaeus proposed a classification. I have already told it is artificial system of classification, but he did not mention any type of evolutionary significance. Same thing in Bentham and Hooker. It is natural system of classification, only based on their phenotypic characters. And first, look into this matter related with evolution, Engler and Painter. So all the students, please noted it. The Engler and Painter is the first pointed that the evolution, any type of classification, when it reflects evolution, it can convert it to phylogenetic classification. And now we already know that is APG4, fully phylogenetic classification, or you can say it is true phylogenetic system. In between that, from artificial to molecular systematics, three taxonomists, renowned taxonomists, Cronquist, Thorne, Dahlgren, proposed a special type of classification. Also, it is called phylogenetic system of classification, or you can say integrated system of classification. Why it is called integrated system of classification? They considered all the characters in plants for final classification. That's why it is called integrated system of classification. And it is so popular in the world. The Conquest Thorne Dahlgren classification. And it's also included in the both postgraduate and undergraduate syllabus. So particularly small plants and grasses, to identify these below generic level on the basis of these characters using a dissecting microscope. Plant species have intra and intraspecific variation in secondary metabolites, which can be utilized as markers, compound for identification and classification of plants. Secondary metabolites are produced as a result of primary metabolism. And the production of these compounds not only involve several genes, but also it is an energy dependent process. Hence, these products cannot be considered as insignificant for the plant and the environment. Modern tools of molecular biology and secondary metabolites present in them can definitely decide about classification of plants. Incorrect identification of plants can be attributed to many problems of resource utilization due to wide availability of modern tools and techniques. Interest has been revived in systematics and current classification of plants based on these parameters for their suitable utilization and resource management. Scientists in this field are now trying to assess the potential of phytochemicals and molecular tools in systematics and classification of plants. So what is actually the taxonomy? Taxonomy is the branch of biology that deals with the identification, place of a new organism into a previously described group, nomenclature, and classification of organism. Plant taxonomy Plant taxonomy is the branch of science which deals with the identification, nomenclature, and classification. It is the basic things of plant taxonomy. Levels of taxonomy. It has 3 levels: alpha taxonomy, beta taxonomy, and gamma taxonomy. Alpha taxonomy, it is concerned with finding, describing, and naming organism. This is actually first and basics stem in taxonomy. So we can say from Linnaeus, Bentham Hooker, all are alpha taxonomists. Beta taxonomy, it includes identification of natural groups and biological classes. This also included in the, we can include in the Linnaeus, Bentham Hooker, and John Ray, etc. Gamma taxonomy, it includes of evolutionary process and patterns. It is included in the phylogenetic systems. Now, I am going to systematics. Biological systematics is the scientific study of the biological diversity and its evolutionary history. Systematics is the classification of living things based on their evolutionary relationship. The science of systematics involves putting living things into categories based on their evolutionary relationship, how closely related they are to each other. Now, taxonomy on the whole, first of all, alpha taxonomy, beta taxonomy, gamma taxonomy. This alpha taxonomy, that is descriptive taxonomy, beta taxonomy and gamma taxonomy, we can say it is phylogenetic taxonomy, and systematics, first taxonomy and after that biosystematics. And these two systematics, we can say it is integrated taxonomy. First one is morphology based taxonomy, second one is molecular based taxonomy, and overall we can say it is e-taxonomy. Again, taxonomy, first part we can be alpha and beta classical taxonomy, typological concept, and omega taxonomy, that is modern taxonomy or neo-systematics or biosystematics. Taxonomy, alpha taxonomy, beta taxonomy, omega taxonomy, cytotaxonomy, chemotaxonomy, and karyotaxonomy. Karyotaxonomy, that is cytological based taxonomy. Today I am discussing only chemotaxonomy, slightly related with molecular taxonomy. Systematics and taxonomy. What is the basic things? John Lindley provided an early definition of systematics in 1830. Although he wrote of systematic botany rather than systematics. Michener et al. 1970 defined systematic biology on taxonomy in relationship to one another as follows. Systematics is the field that provides scientific names for organisms, describe them, preserve collection of them, provide classification of the organism. On the other hand, taxonomy is the part of systematics concerned with all the above topics of systematics. In this chart, I have, that is markers. What is called markers? That is identifying characters, attributes, we can say it is markers. So first of all, morphological markers. I have already discussed about Linnaeus, Bentham Hooker, John Ray, Hutchinson, all are classification based on morphological based. And second one is biochemical based, and now molecular based. So we are today's lecture mainly concerned with biochemical, that is protein binding patterns, isozymes, and flavonoid, few secondary metabolites, and so on. So now coming to my main topic, that is chemotaxonomy. What is actually the chemotaxonomy? It's the definition of chemotaxonomy. This system of classification relies on the chemical similarity of a taxon. That is, it is based on the existence of relationship between constituents in various plants. It is the latest system of classification that gives more scope for understanding the relationship between chemical constituents, their biosynthesis, and their possible action. It involves study of the chemical variation in a diversity of organisms and their relationship. So, the classification of plants on the basis or based on chemical content is called chemotaxonomy or chemosystematics or chemical taxonomy. So this definition proposed by Professor Naik from Aurangabad University. Data from chemistry of plant products is termed phytochemistry. The principles, procedures, and result of investigations of chemical variation of plant groups can be applied mainly for two purposes. Number one, to provide taxonomic characters which may help in plant classification and may improve the existing system of plant classification. And number two, to have additional knowledge of phylogeny or evolutionary relationship of plants. So what is the purpose of chemotaxonomy? The evidence is used in classification of plants into two main purposes. Number one, to improve the existing system of plant classification. And number two, to develop the present day knowledge of natural relationship of plants. Three broad categories of compounds are used in chemotaxonomy. Number one, primary metabolites. Number two, secondary metabolites. And number three, Sementides. My title also reflects chemotaxonomy, its application in systematics, plant systematics, with special emphasis to Sementides. So primary metabolites are parts of vital metabolic pathways. Most of them are of universal occurrence and is utilized by the plant itself for growth and development. Examples: starch, chlorophyll, alluron grains, citric acid, aconitic acid, etc. Secondary metabolites. Secondary metabolites present in plants used for for protection and defense. Example: glycoside, alkaloids, volatile oils, flavonoids, anthocyanin, betaline, plant phenolics, etc. And sementides, these are the important characters: proteins, amino acids, DNA, RNA, etc. So where can we use this technique? কোথায় কোথায় আমরা এটা ব্যবহার করতে পারি? Chemotaxonomy has been used in all the groups of plant kingdom, such as fungi and bacteria, algae, bryophytes, pteridophytes, gymnosperms, and angiosperms, starting from the rank of variety up to the rank of division. Chemical characters are particularly of high taxonomic value when they are stable, unambiguous, and not easily, if at all, changeable. Stages in chemotaxonomic investigation. Hognier, 1967, recognized 6 chief categories in a phytochemical study. First one, choice of a group, taxonomic survey, and sound sampling. This is very important. Choice of target, choice of samples, choice, mastery, and modification of suitable chemical techniques in a pilot survey. Third, full analysis of all material. Fourth, interpretation and comparison with data from all other sources. Number 5, adjustment of classification as necessary. And number 6, treatment of any evolutionary relationship as indicated by the new data. Phytochemical characters of taxonomic significance. First, that is the occurrence and distribution of the various types of chemical substances present in plant proved to be of taxonomic significance. Second, it should be noted that all kinds of chemical substances present in plants do not reveal information useful to the taxonomist. According to Menzer, 1966, phytochemical characters of taxonomic significance have been classified into 3 types: primary constituents, secondary constituents, and miscellaneous substances. We already know that is primary constituents, secondary constituents, but miscellaneous substances like plant producing wax, these types of substances we can categorized under the miscellaneous substances. The 3 kinds of components: primary metabolites, secondary, I have already told this, primary metabolites, secondary metabolites, and Sementides. Primary constituents, these includes macromolecular compounds directly taking part in metabolism and include proteins, nucleic acid, chlorophyll, and polysaccharides. Number two, all chemical materials synthesized by an organism reflect the information in DNA, RNA, and protein. Third point, DNA, RNA, and proteins molecules have been termed as sementides. Sementides thus contain useful information of taxonomy and phylogeny. Secondary constituents, they include compound lacking nitrogen and not involved directly in plant metabolism. That is, simple phenolic compounds like caffeic, benzoic, and nicotinic acid, polyphenolic compound like flavonoids, terpenes, coumarins, alkaloids, and pigments of which flavonoids are most widely studied with respect to plant systematics. The phenolics, alkaloids, terpenoids, and non-protein amino acids are the four important and widely exploited group of compound utilized for chemical classification. This group of compounds exhibit a wide variation in chemical diversity, distribution, and functions. The chemical constituents of plants differ from species to species, restricted to certain taxa. Thus, they are the valuable characters for plant classification. Jones and Luxinger is a renowned systematist. In 1987, classified phytochemical characters into two major groups on the basis of their molecular weight, which are useful in taxonomy. First, micromolecules. Second, macromolecules. Low molecular weight compound with a molecular weight of 1,000 or less. Example, amino acid, alkaloid, fatty acid, terpenoid, flavonoid. These are the micromolecules. Now, high molecular weight compound with a molecular weight of over 1,000. Example, proteins, DNA, RNA, complex of polysaccharides. We can say this macromolecules. It's more scientific than previous all the definitions. Micromolecules, macromolecules, based on their molecular size. First one is proteins. Among the various sementides, proteins serve as the most important tool in chemotaxonomy. The importance of proteins in chemotaxonomy has several reasons behind. First, these are large complex molecules showing little quantitative variation with changing environmental factors. Secondly, they are universally distributed. Like in the plant level of view, that is Asteraceae family is a universally or cosmopolitan in distribution. Protein also has universally distributed and present. Third, they are alternatively simple to extract and handle and present in appreciable amount. And the last, numerous cheap, simple, and rapid methods have developed in the recent past of protein analysis and comparison of taxonomists, of which electrophoresis is particularly an important method of protein separation. Comparison of the protein banding patterns. In recent times, protein band patterns obtained by gel electrophoresis have been focused on the plant identification of critical taxa, their relationship and taxonomic status. Taxonomic interpretation should be based on comparison of proteins from homologous organs of the same age in order to avoid any confusion due to variability of proteins. Considerable variation in protein complements has been recorded at the level of species and genus and even between the same plant in different populations. This form evidence upon which taxonomic systems may be funded, tested, or demolished. This is actually the SDS page, protein profile for comparative study and protein banding pattern. This is actually diagrammatic protein banding pattern. And banding patterns, bands common to species A and B is divided by total bands in species A and B. With the help of numerical taxonomy, we can form a data matrix, and with the help of numerical taxonomy, form a dendrogram. And we can easily say it is high percentage of similarity, how it could be distantly related or closely related. Within the genus, within the family, and different species in a family. Why can we use electrophoresis technique in chemotaxonomic studies, particularly in protein investigation? We should know ABC of gel electrophoresis. Because of the presence of ionizable molecules on the surface of proteins, they will migrate when subjected to an electrical field in a solution of suitable pH. Soluble proteins thus migrate within an electric gradient at a rate that depends on their neat electric charge and on their molecular size and shape. The rate of migration of each protein is constant under identical condition and hence can be used as a reliable character for the detection of homologous protein. The diagrammatic figure in the left, entire gel electrophoresis system with gel, and each and every part I have detected in the labeling form. So you can say also, you can see this is electrode, this one is buffer, this one is high molecular weight protein, this is low molecular weight protein, this is electrode, and so on. An overview of SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. This is actually the diagrammatic figure of two gel system, that is stacking gel and resolving gel. The two gel pH is totally different, 6.8 for stacking gel and resolving gel 8.8 to 8.9. This is cathode, this is anode. The direction of electrophoresis, the mixture of macromolecules with the help of electrophoresis, porous gel, and finally we get the result. It has been possible to obtain useful systematic information from electrophoretic analysis of crude biological protein sample. For example, Johnson and Hall, 1965, have demonstrated the phylogenetic affinities in trypticine by the process of protein electrophoresis. This process also helps in establishing a close relationship between Vicia and Lithyrus. In my PhD, we have also studied with the help of gel electrophoresis interspecific variation among 8 species of Cassia, Cisalpinaceae, and in the postdoc Rixia in the Darjeeling Government College were evaluated on the basis of seed protein, spore protein, thallus protein, and mitochondrial DNA RFLP by gel electrophoresis. This is our publication, Mondal et al. 2000 and 2007, as well as pollen protein patterns, Mondal et al. 1998. This is actually our photograph from my thesis. The the protein profile of the seeds of Phaseolus vulgaris as revealed by gel electrophoresis, the protein profile of the pollen of Cassia siamia as evaluated by gel electrophoresis. And the second is amino acid sequence studied of homologous proteins. Comparison of amino acid sequences of homologous proteins from different taxa has also provided a powerful tool for evolutionary and systematic studies. Our first publication, myself, Professor Sanjukta Mondal Parui, and my supervisor, Professor Sudhendu Mondal, published a remarkable paper in the current science, free amino acids of 46 angiospermic taxa from pollen we have to use for marker. So free amino acid used as a marker in the field of taxonomy. This is the first publication in current science, 15 pages publication. It is free and you can download it and see it for your future research also. The degree of similarity in the amino acid sequence is presumably proportional to the degree of genetic relationship. Professor Sivarajan know that is a good book and is inspiring me. I am also inspired from his book, Sivarajan, Plant Taxonomy. It's a good book on the theoretical point of view. This is the photograph, tells thin layer chromatography of free amino acid of the pollen of the several species of plants. This is from my thesis. And this is also paper chromatography. It's also obtained from my thesis. Third one is analysis of isozymes and allozymes. Electrophoresis of enzymes can reveal two distinctively genetically controlled variation in enzymes phenotypes. First one is allozymes. They are different forms of a particular enzyme that are coded by a single gene locus, but by more than one different allele, each coding for a slightly different amino acid sequence which have difference in mobility during electrophoresis. Second, isoenzymes. They are different forms of the same enzyme formed from genes at different loci. This is actually different types of isoform, isozymes, banding patterns. Isozymes are genetically controlled variants of enzymes which have various metabolic functions in plants distinction between isozymes, homologous proteins coded by different loci, and allozymes, allelic variants of a particular protein formed at the same locus, allows us to examine the alleles at loci without doing crossing studies. Procedures: number 1, enzyme extracted from the plant. Number 2, separated via electrophoresis. Third, get bands of proteins that reflect isozyme profile. Isozyme have been most useful at the species level and below, have been very useful to examine complex cases of hybridization and reticulate evolution because they are additive. Examples have shown that hexaploid bred wheat Triticum aestivum contains some of proteins possessed by ancestral species Triticum diocum, a tetraploid, plus Agileops quarosa, a diploid. This picture also from my thesis, peroxidase isozyme profile of the leaves of some plants, peroxidase isozyme profiles of leaves of some plants. Isozymes, allozymes, electrophoresis system. Previously, I have told regarding SDS page. This is actually slab gel, horizontal gel for enzyme system. This is actually the enzyme systems, and this is actually the banding patterns. This work from is my postdoctoral work, different species of Rixia and Markensia, worked during my Darjeeling Government College and in a major project UGC. This is also the peroxidase, esterase, and acid phosphatase, different types of isoenzymes in different forms. Nowadays, a large number of such different enzymes are being exploited electrophoretically for taxonomic purposes, particularly to construct phylogenetic either within or between species. Most of these include enzymes involved in fundamental cellular process such as those involved in glycolysis, hexokinase, phosphofructokinase, aldolase, etc., Krebs cycles, isocitrate dehydrogenase, malate dehydrogenase, etc., enzymes involved in protecting cells from free radicals, catalase, superoxidase, and superoxide dismutase. Comparative studies of enzymes may yield data about differences in primary structure which can be traced to evolutionary adaptations. For example, Natarella and in 1975 studied the peroxidases and proteins of 4 species of the genus Petunia, Petunia axillaris, Petunia inflata, Petunia violacea, and Petunia poradi, and other 11 cultivars of Petunia hybrida by gel electrophoresis. Electrophoretic patterns reveal that all the species are closely related and that Petunia inflata, Petunia axillaris, and Petunia violacea are involved in Petunia hybrida. Similarly, based on peroxidase, esterase, and acid phosphatase isozymes pattern, Chemonoids and Sequoids 1984 suggested that the genus Teninantherum, formerly considered as a part of the genus Hodam, should be treated as an independent genus. So taxonomic delimitation, inclusion is possible with the help of binding patterns both in the protein levels, isoenzymes level, and amino acid level. What is the basic difference between allozymes and isozymes? Allozymes are multiple forms of an enzyme that are coded by the different alleles at the same locus. And on the isozymes are multiple forms of an enzyme that are coded by the different genes and different loci. Second point, different alleles at the same locus in allozymes, but genes at different loci in isoenzymes. A number of genes involved, a single gene involved in allozymes, and several genes are involved in isoenzymes. And allozymes, all the same locus in case of alleles, in case of isoenzymes, have different loci. So what is the advantages of allozymes and isozymes? Co-dominant type markers, easy to use, easily adjustable from species to species, low cost, and general and general applicability. Disadvantages of allozymes and isozymes: limited number of available allozyme loci precludes their use in large-scale genome mapping. Number two, heterozygote deficiencies due to null alleles. Number three, reduce the level of detectable variation due to some changes in DNA sequence. And number four, only a small fraction of enzyme loci appear to be allozymetically polymorphic. Concluding remarks on isoenzymes: although isozymes, allozymes are not used so much, but yet it gives satisfactory result. It should apply to the genetics of phylogeny as it gives good result. It has limited use in fisheries science rather than other sciences. It requires good, adequate infrastructure, particularly 4-degree temperature laboratory, constant with molecular equipments. It's possible to use isozymes and allozymes for the population studies also. And now I'm going to my special emphasis part, that is Sementides. The term Sementides and related term coined by Linus Pauling and Emil Zuckerman. Sementides are information carrying molecules. Sementides provide taxonomic data not on the basis of presence or absence, but in terms of sequences, ratios, or percentage. Sementides or sematophoric molecules are biological macromolecules that carry genetic information or a transcript thereof. 3 different categories or sementides are distinguished. Primary sementides are genes which consist of DNA. Secondary sementides are messenger RNA, which are transcribed from DNA. And third, tertiary Sementides are polypeptides, which are translated from messenger RNA. In eukaryotic organism, primary Sementides may consist of molecular, nuclear, mitochondrial, or plastid DNA. Not all primary Sementides, ultimately from tertiary Sementides. Some primary Sementides are not transcribed into messenger RNA, that is called non-coding DNA, and some secondary Sementides are not translated into polypeptides, that is non-coding RNA. The complexity of Sementides varies greatly. For tertiary Sementides, large globular polypeptide chain are most complex, while structural proteins consisting of repeating simple sequences are least complex. Although Sementides are the major types of data used in modern phylogenetics, the term itself is not commonly used. This is actually the DNA, RNA, and protein transcription, translation, and finally protein. Types of protein structure: primary structure, secondary structure, tertiary structure, and quaternary structure of proteins. First, isosemantic DNA or RNA that differ in base sequence but translate into identical polypeptide chains are referred to as being isosemantic. Episemantic molecules that are synthesized by enzymes are referred to as episemantic molecules. Episemantic molecules have a larger variety in types than Sementides, which only consist of three types: DNA, RNA, or polypeptides. Not all polypeptides are tertiary Sementides. Some mainly small polypeptides can also be episemantic molecules. And the last one is asemantic. Molecules that are not produced by an organism are referred to as asymmetric molecules because they do not contain any genes information. Asymmetric molecules may be changed into episymmetric molecules by anabolic process. Asymmetric molecules may also become symmetric molecules when they integrate into a genome. Example of certain viruses and episomes have this ability. When referring to a molecule as being symmetric, episymmetric, or asymmetric, then these only apply to a specific organism. Asymmetric molecules for one organism may be asymmetric for another organism. So primary Sementide A, DNA, primary Sementide B, DNA, secondary Sementide Q, messenger RNA, secondary Sementide P, also from messenger RNA, tertiary Sementide, it is actually polypeptide, and asymmetric compound anabolism, that is episymmetric molecules. Application of Sementides. Sementides are used as phylogenetic information for studying the evolutionary history of organisms. Primary Sementides are used are also used in comparative biodiversity analysis. Why it is applied in the biodiversity? We know that the diversity of organism or genetical variation in a particular area, we can say it is biodiversity. So we can use these Sementides for biodiversity analysis. However, since extracellular DNA can persist for some time, this type of analysis cannot discern active from inactive and or dead organism. The extent to which biological macromolecules are informative for studying evolutionary history differs. The more complex a molecule, the more informative it is in for phylogenetic. Primary and secondary Sementides contain the most information. In tertiary Sementides, some information is lost because many amino acids are coded for by more than one codon. Episementic molecules, example carotenoids, are also informative for phylogenetic. However, the distribution of these molecules correlate preferably with phylogenesis based on Sementides. Therefore, dependent confirmation is often still needed. The more enzymes involved in a synthesis pathway, the more unlikely that such pathways have evolved separately. Therefore, we can say for episementic molecules, molecules that are synthesized from the least complex asymmetric molecules are the most informative in phylogenetics. In few exceptional cases, however, different pathways may synthesize similar or even identical molecules. For example, in animals, plants, and other eukaryotes, different pathways have been found for vitamin C synthesis. Therefore, certain molecules should not be used for studying phylogenetic relationship. Limitation of Sementides. Although asymmetric molecules could indicate some quantitative or qualitative features of a group of organisms, they are considered to be unreliable or uninformative for phylogenetics. Analysis using different Sementides may yield conflicting phylogenesis. However, if the phylogenesis are congruent, then there is more support for the evolutionary relationship. By analyzing larger sequences, complete mitochondrial genome sequences, phylogenesis can be constructed which are more resolved and have more support. Secondary metabolites, phenolics have been the most frequent used in taxonomy, especially flavonoids. Example, anthocyanin and betalains. Anthocyanins are flavonoids with two sugar group. Betalains have heterocyclic nitrogen containing aromatic rings, mutually exclusive different metabolic pathways. Phenolics, derivatives of phenolic compounds. Plants are classified on the basis of specific phenolic compounds. Example, leucoanthocyanin, abundant in woody plants, flavonoids and methoxycyanomatic acid in herbaceous plants, ellagic acid in tribe Caria of Rosaceae, isoflavone iridine in section of pagovinin of iris. Absence of iridine, iris flavonimoma was removed from this section. This is actually the phenols classification. Plant phenols, first simple benzoic acid derivatives, cinnamamic acid derivative. In case of complex, flavonoids, tannins, stevins. Flavonoids have three parts: flavonoids, flavins, and anthocyanins. Classifications of phenolics: phenolic compound, polyphenols, simple phenols, tannins, and flavonoids. Simple phenolics, phenolic acid, coumarins. Phenolic acids again divided into two: hydroxybenzoic acid, hydroxycinnamamic acid. Betalins. Betalins are derived of phenols serving as pigments. They are present in 10 families of angiosperms. The position of cactaceae was disputed for many years. On the basis of presence of betalin, the position in centospermi is confirmed. Cactaceae also confirmed with the help of betalins, pigment or characters. Beta vulgaris from betalins divided into two: beta cyanins, beta xanthins. Beta cyanins again divided into betaine and isobetaine, and beta xanthins divided into three: valgaxanthinin 1 and 2, and in incaxanthin. This is the source of betalin, natural source, one from cactus and from Beta vulgaris, and also from anthocyanin. This is the natural source of anthocyanin. Anthocyanins have different forms: cyanidin, malvidin, plagiarodin, putidin, peonidin, and delphinidin. All are active and are edible and are very useful in human being, particularly during this COVID time. We can consume more anthocyanin. It has the anti-cancer, anti-virus, different types of, and our body will immune system will develop with the help of anthocyanin, betanin, these types of natural product. And we know that plant is only the producer of these types of antioxidants. Alkaloids are compounds containing heterocyclic nitrogen basic characters and complex molecule structure. Such compounds are restricted to phylogenetics, about 5,000 alkaloids in angiosperms. Alkaloids are divided into three: true alkaloids, protoalkaloids, pseudoalkaloids. First, true alkaloids have a nitrogen containing nucleus derived from biogenetic amine. Protoalkaloids derived from amino acids but lack a heterocyclic ring. They are usually simple amines, example ephedrine, mescaline. Sometimes they may be precursor of true alkaloids. And pseudoalkaloids biologically unrelated to amino acids. Most of them derived from terpenes, steroids, nicotinic acid, or purines. Classification of alkaloids: true alkaloids I have already discussed, that are derived from amino acid and have nitrogen in a heterocyclic ring, example atropine. Protoalkaloids that are derived from amino acid and do not have nitrogen in a heterocyclic ring, ephedrine. Pseudoalkaloids that are not derived from amino acid but have nitrogen in a heterocyclic ring, caffeine, example caffeine. And false alkaloids are non-alkaloids give false positive reaction with alkaloidal reagent. Alkaloids are more or less toxic substances which act primarily on CNS. They have a basic characters, contain heterocyclic nitrogen and synthesized in plants from amino acid and or their immediate derivatives. They are of limited distribution in plant kingdom, but such definition exclude a number of nitrogen containing plant constituents which are normally thought as alkaloids. Biological amines like ephedrine, hardenine, betaine, choline, muscarine are commonly examples. All such alkaloids are separately referred as protoalkaloids or biological amines or aminoalkaloids. In plants, alkaloids, due to their basic nature, generally exist as a salt or organic acid like oxalic acid, citric acid, malic acid, tartaric acid, tannic acid, etc. Some alkaloids like narcine and nicotine are occurring free in nature. A few alkaloids occur as glycoside of sugars like glucose, rhamnose, and galactose. Example alkaloids of solanum and veratrum, group of amides, piperine as esters, atropine and cocaine of organic acids. But when protoalkaloids and true alkaloids occur in same genus of family, both are usually classified as alkaloids. Example, hardenine, muscaline in Cactaceae family. Another group of nitrogen compounds that fall outside our definition of alkaloids are mono, diterpenes, steroids, nicotinic acid, purines. These can also be classified according to precursor on which they are based and collectively called as pseudoalkaloids or alkaloid interfacta. Example, cosine and caffeine. Alkaloids in plants, there is no sharp dividing line between alkaloids containing plants and alkaloids free plants. From practical point of view, we can define lower limit as 0.01% of dry weight. Another characteristics of true alkaloid is that it always contains more than one alkaloids. We can cite example, a ricine is only alkaloids in Ricinus communis. You know that these plants belonging to the family Euphorbiaceae and it's very common, and Jansianaceae, it's not very common, only alkaloids is Jansianaceae. That's why these two bases have been kept outside the class of true alkaloids. Occurrence and distributions. Flavonoids. Flavonoids are more abundant in hard plants, particularly in the Magnoliopsida, especially in the angiosperm families. Apocynaceae, Papaver, Ranunculaceae, Rubiaceae, Rutaceae, Solanaceae, but less frequent in lower plants like algae and bryophytes, pteridophytes, and fungi. This is actually the flavonoids other subgroups like anthocyanidin, flavonoids, flavonols, isoflavone, flavones, and flavonoids. Natural source of flavonoids. We are the lucky enough botany student. All the chemicals, phytochemicals those which are directly related with human being for their protection immunity are sourced from plant source. And last part, it is not directly related with chemotaxonomy, but when I am emphasizing the that is Sementides, it is a part of DNA. So big explosion in last 10 years in this kind of data, 3 genomes in plants. Just I am mentioning the not in detail because DNA is a 3 or 4 lectures is required for student in the phylogenetic point of view. But today I am just discussing the basic things and application to a thing. Big explosion in last 10 years in this kind of data, 3 genomes we have considered: chloroplast, mitochondrial, and nuclear. In my postgraduate classes, I have deliberately told in my classes, both special and general, molecular taxonomy, and I have mentioned in detail PhD coursework classes also, the chloroplast DNA, mitochondrial DNA, and nuclear DNA. Chloroplast have been used most often. Mitochondrial genome has structural rearrangement, making it hard to interpret. Nuclear gene often have many copies of gene as well as concerned evolution. RFLP analysis, it is a very common method in DNA analysis. Restriction enzyme cleave DNA at very specific point, particularly in the phylogenetic study. Many such enzymes are known, each cleaving at different recognition site, nucleotide sequences. These produce fragments of different lengths which can be separated via gel electrophoresis and hybridized to radioactive probes to determine where on the chloroplast genome they are located. Maps of the restriction sites are then constructed. We can detect numerous kinds of changes, including point mutation, inversions, and deletion. These can be used in cladistic analysis. Example, in asteroids. DNA sequencing is now practical to look directly at the sequence of nucleotide to get data. We know that our APG system only based on DNA sequencing, particularly cpDNA, nuclear DNA. Used PCR to obtain many copies of a gene, usually use automated sequencer. PCR, we know that polymerase chain reaction consists of 3 steps: denaturation, annealing, and extension. In graphically, the structure of DNA, the PCR component, DNA samples use primer, tag polymerase, mixing buffer, nucleotide, a PCR tube in a thermocycler machine, that is PCR machine, and first denaturing, after annealing and extension of, it is actually the variation of temperature. First, that is denaturation with the help of 95 degrees centigrade. After that, annealing 55 degrees centigrade, primer bind template, and extension, that is 72 degree, synthesis new strand with the help of 72 degree centigrade. The chemotaxonomy or molecular systematics, broadly we can say chemotaxonomy, and partly we can say molecular systematics, can use chemical molecular characters to study relationship at any level in the hierarchy: population, species, genera, families. In our laboratory, Vidyasagar University laboratory, plant taxonomy, biosystematic and molecular taxonomy laboratory, we are commonly use these chemotaxonomy and molecular systematics methods and application, and we have already 46 species detected rare endangered medicinal plants from Southwest Bengal. With the help of my total teamwork, research team, we have detected with the help of DNA barcoding. It's a separate chapter, and already deposited our data in the NCBI, and we got the final accession number in the worldwide reputation in our institute. So it is a great achievement in the Vidyasagar University Department of Botany and Forestry with the help of molecular systematics. Chemical and molecular characters can be used in cladistic analysis to analyze phylogenetic relationship, can be used to analyze variation within species. Now 10 research scholars are there working in different fields. One of my students, Mumtaz Khatun, working in the sclerites, is a dead cell of plant tissue, and there are ultrastructural studies in different families of angiosperms. Arjun Patro working on the two aspects, on the antimicrobial aspects and on the floristic points of view of Purba Mednipur, few mangrove areas, on the family Acanthaceae. One of my students, Amal Mondal, working on the cytotaxonomical with the DNA, with the help of few DNA barcoding of hemiparasitic taxa. One of my scholar, Oyan Noskor, working on the medicinal plants of Southwest Bengal based on DNA barcoding data. He is now working under the DRS SAP2 fellow. One of my research scholar, Segrosidul Islam, working in the first time in West Bengal, that is liverworts or brophyte flora of Southwest Bengal, with special emphasis to molecular systematics. Another students, the Anup Bhuya, working in the natural fiber. He comes from Sobong, and we know that Mednipur is a biologically hotspot place, and this natural fiber, particularly in Sobong, we know that there's a Madhur plant, that is Cyperus pangauri. 2 or 3 students already been submitted their thesis, like Sibdas Maiti already joined in a college, and one of my students jointly worked with remote sensing department. He also worked on the Jungle Mahal and in mangrove areas with the help of GIS. And Sibdas' work is interesting on the point of view, that is Golaghat thermal power pollution with the help of geospatial technology. And another few scholars working under my guidance in different aspects of systematics, chemotaxonomy, molecular systematics. And few students already been awarded. One of they are submitted and awarded from my guidance. So this is a remarkable work in different fields of plant systematics from my laboratory, from my department and my university. If any doubt, you can consult my one textbook, Advanced Plant Taxonomy. It's a good, not my book, it is a good book on the sense all the informations are practical information included in my book. All the photographs, all the information, all the research documents from my thesis, from our research. So you can consult particularly chemotaxonomy part or other parts also, but this complex chapter is simply included in my books also. So please consider. And lastly, thank you to all to listen, hearing my lecture.
Hello. Hello.
Thank you, sir. Hello.
Hello. I would like to give special thanks to Professor Mondal because the book written by sir is very comfortable and affordable and knowledgeable because the present graduate syllabus, undergraduate syllabus is based on the UGC core syllabus. That means in all university has used the UGC core syllabus, and the taxonomy topic or paper in the UGC undergraduate syllabus is based on molecular taxonomy or different aspect, is very much relevant to the book which is written by Professor Mondal. Thank you, sir. And this book also helps the undergraduate students very strongly and in all aspects. Thank you, sir, for this book.
Thank you. Welcome.
I think Vishwajit Babu have a question.
Yes.
Sir,
Sir.
please, you directly ask, sir.
Yes, Biswajit, কেমন আছো? How are you?
হ্যাঁ স্যার ভালো আছি আপনি ভালো আছেন তো স্যার?
হ্যাঁ, এই যাবত তো আমার অন্য লেকচার শুনেছো, আজকে একটু অন্য ধরনের লেকচার দিলাম।
হ্যাঁ স্যার খুব ভালো লেগেছে। হ্যাঁ স্যার একটা জিনিস আপনার ওইখানে পয়েন্টে দেখলাম, ওই আইসোএনজাইমের ডিসঅ্যাডভান্টেজে নাল অ্যালিল টার্মটা দেখেছি। ওই নাল অ্যালিলটা একটু যদি আমাকে বলেন।
নাল অ্যালিল,
হ্যাঁ,
যেখান থেকে এই এনজাইমটা ডিটেক্ট হচ্ছে না তাকে আমরা নাল অ্যালিল বলতে পারি। কারণ এখানে তো সব জায়গা থেকে হয় না।
এটা কোন পার্ট, কি পার্টটা সেটা?
ধরো এনজাইম আর আইসোএনজাইম, তাই তো?
ইয়েস।
ঠিক আছে, আইসোএনজাইম হচ্ছে ডিফারেন্ট ফর্মস কিন্তু ফাংশন এক, আর অ্যালোজাইমটা হচ্ছে নির্দিষ্ট অ্যালিল থেকে আমার সেই ইয়েটা হচ্ছে, তা যেখান থেকে হচ্ছে না তাকে আমরা নান বলছি।
আর স্যার লিউকোঅ্যান্থোসায়ানিন আপনি বললেন উডি প্ল্যান্ট।
হ্যাঁ,
ওটা
দেখবে একটা এক্সাম্পল, তুমি তো গ্রামের ছেলে, একটা জাস্ট ফর এক্সাম্পল আমার চোখের সামনে এটা ঘটলো। ধরো তুমি যদি পেল্টোফোরাম গাছকে কেটে রেখে দাও, এটা তো উডি গাছ একটা, তাহলে দেখবে একদিন আবার দুদিন পরে দেখবে তার স্টেমের কালারটা চেঞ্জ হয়ে গেল।
আচ্ছা,
দিস ইজ কল্ড, যে টার্মটা ব্যবহার করলাম এইটাই উডের ক্ষেত্রে পাওয়া যায়।
মানে যে কোনো উডি প্ল্যান্টে,
যে কোনো উডি প্ল্যান্ট নয়, সবার ক্ষেত্রে এটা ঘটে না। ঠিক আছে, একটা এক্সাম্পল দিলাম, পেল্টোফোরাম এনার্মি।
আচ্ছা স্যার,
তুমি যখন শুনে নাও, যখন তুমি দেখবে এটা কাটা হচ্ছে একটা কালার, কিন্তু কেটে রাখার পরে দেখবে কালারটা চেঞ্জ হয়ে যাচ্ছে, তখনই ধরা হয় যে কাদের মধ্যে আছে কাদের মধ্যে নেই।
Sir, sir, sorry to interrupt, sir.
Yes, sir. Mani, কেমন আছো?
স্যার ভালো আছি।
আচ্ছা।
আমি বলছি স্যার অনেকেই তো আছে আউটসাইড স্টেট, স্যার একটু ওরা রিকোয়েস্ট করছে ইংলিশে একটু বলতে আর কি।
আমি তো তাই চাইছিলাম, তোমরাই তো বললে দুরকম ভাষায় বলতে হবে।
না না অনেকে রিকোয়েস্ট
ইংলিশেই
করছে।
বলুন স্যার।
হ্যাঁ স্যার
ঠিক আছে।
আচ্ছা স্যার,
ঠিক আছে।
অ্যান্থোসায়ানিন ইজ অ্যান্টিভাইরাস অ্যাক্টিভিটিস।
আচ্ছা মানে প্লিজ অন ইওর ভিডিও
ওকে
and talk.
Anthocyanin
Okay, sir.
activities is antivirus. So which types of virus? Please.
No. This COVID situation, my previous lecture on in your college on ashwagandha. So ashwagandha not produced any type of anthocyanin, but my observation, my work is that this type of pigment can enhance your immunity. And during this COVID period, in daily newspaper, in internet or any type of research paper, all the doctors, all the physicians told that you can enhance your immunity power. Then we can fight the COVID-19. That's why I am said this.
One question, sir.
Yes.
How to extract enzyme from the plants, and how to identify this enzyme?
Oh, very good questions. Very good question, but it is a long procedure. You collect fresh sample or any tip portion of that is immature part of plant, okay, and fresh, not in preserve, with the help of buffer. Either it is sulfate phosphate buffer or it is HCl buffer. I homogenize, and with the help of centrifugation, 5,000 to 10,000 4-degree temperature. Okay, extra centrifuge and supernated collection of supernation for gel electrophoresis, and with the help of particular substrate, you are use esterase, peroxidase, acid phosphatase. These three isoenzymes have different have specific substrate. So you have to first of all purchase from Sigma of this substrate, and after that we can use commonly in our laboratory for detection of particular enzyme. But mind it that enzyme is not in a gel, it's not a long time visible, it is a short time visible. When it is visible, please take your photograph, and after few minutes it is disappear. In case of free amino acid, you also start with thin layer chromatography with the help of TLC sheet. But when it is with the help of ninhydrin application, when it is evolved, just take photograph. After that you can analyze. Just after 30 to 1 hour, it is removed from your TLC sheet, or in case of enzyme, it is removed from your gel, or it is diffused. But in case of protein gel, when you stain with chromasic brilliant blue, it is sustainable. It is persistent. You can save it several months also, or you can put in a gel dock system for particular each and every band protein content, protein quality, and after that with the help of numerical taxonomy you evaluate with the data matrix and form a cladogram, dendrogram, whatever may be your application.
Sir, I understood my confusion.
Thank you.
I request, মানে আমার এটা বলা ঠিক হবে কিনা আমি জানি না এইখানে, তবু আমি আমার আবেগ থেকে বলছি স্যার, যদি সম্ভব হয় দেখবেন লাস্টে কোশ্চেন ইন্টারাকশনের পরে একটা আপনি যদি গান শোনান, যদি মমতাজ অর্জুন এরা পারমিট করে, জানি না, আমি মানে ফেসবুকে একটা গান শুনলাম, যদি একটা করেন বোধহয় এই কোভিড পিরিয়ডে আমরা তো সব হ্যাঙ্গিং অবস্থায় আছি, এটা মনটা ভালো লাগবে আপনার কাছে যদি পাই, যদি সবাই পারমিট করে। থ্যাংক ইউ স্যার।
Thank you. Welcome.
Another question in chat box from sir.
Yes, yes.
Yes, sir. Another question in chat box from Nitya Papuraj.
Just a minute.
I inform Nitya Papuraj, you can directly question to our resource person. Nitya
Yes,
Papuraj, you can directly interact.
Thank you, sir. Your lecture was very good. Sir, can you hear me?
Thank you, ma'am. Yes, hear me.
Sir, how many plants,
Please,
that is
please
quantity,
fast
how
open
many
your.
plants will be needed to
Nita, listen to me. Please open your video first.
Sir, I am not in formal dress. That's why I am not open the video.
Carry on.
Thank you, sir.
Your audio is
Sir,
disturbing.
your presentation was very good.
Thank you.
Just a minute, sir. Just a minute, sir. Sir, can you hear me?
Yes, hear me.
Sir, I have one question. How many plants will be needed to engage in this chemotaxonomic research? Because so many researchers, they are unknowingly collecting some plants like kgs. They are abundantly collecting plants. So I need that. How much plant is needed for this chemotaxonomic research?
How many plants?
Yes, yes.
It depends upon your research.
Some plants may be under threat condition, no? So if you tell me about the quantity, that will be easy for that further researchers, right? So that's why I'm asking this question.
In case of solving the taxonomical problem, you choose, suppose example, from Asteraceae family. You have to take, because we know that the Asteraceae have cosmopolitan in distribution. You minimum you collect 25 to 30 plants for numerical taxonomy also, because we are the final output with the help of numerical taxonomy. We know that it is a separate chapter, but it is closely related with chemotaxonomy, molecular taxonomy, and finally numerical taxonomy. So any type of taxonomical dispute, you should select according to your problem, either 10 species, either 5 species, or 100 species. In my postdoc work in the Darjeeling Government College, I have taken all the Martensialis group for chemotaxonomy and molecular taxonomy. So enough data or enough species you can analyze, but with the help of sophisticated hands and techniques and your equipment. Without any type of disputation, it's not possible to a better result. Mind it.
Okay, sir. Okay, sir. Thank you, sir.
Welcome. You are from which place?
I am from Tamil Nadu, Trichy, sir.
Okay. Nice to meet you.
Thank you so much, sir.
Okay.
Sir, another question from a participant from YouTube box, chat box. So if you kindly tell about numerical taxonomy in brief.
No. It's a big chapter. It's not possible in a one or two word. You know that, Arjun. It's so difficult than molecule, chemo, after that numerical. And in my class also, both postgraduate, undergraduate, and PhD, without numerical taxonomy, we can't move in a good systematic way. You know that. So it's not possible one or two words or numerical taxonomy or taximetrics based on mathematics and computer with the help of biological data attributes. So please.
Sir, as as a botany student, we all know about the ICBN or IBC, and
Yes.
you have already joined
Yes,
previously.
attended. Yes, I am the lucky.
Attended IBC. So if you kindly share your some experiences from this
Yes.
Melbourne court.
Yes. Good question. Not related my topic, today's topic. It is a overwhelmed.
I think that will favor to all students.
It is overwhelmed experience. When I was in MSc student, first taught about conquest thorn Dahlgren from my teacher, Professor Prashanto Bhattacharya, and several lecture on ICBN from myself. And when I joined in the Vishwabharati for PhD after qualifying CSIR under supervisor Professor Sudhendu Mondal. So he first joined, you know that Tokyo is also one court in 1993. He joined, and when he came back, explained all the ICBN dignitaries like Thorne, Dahlgren, and Conquest. So I am so inspired. Well, it could be possible to me join as a participant or as a delegates or any type of invitation in any ICBN. So my work, you know that, not highlighted in your introduction, mainly all the parts cover only one part, that is hemiparasitic taxa. It is already established from Vidyasagar University, my lab. 4 or 5 students already been got their PhD from this area. So since my postgraduate level, interest on that this subject, and unfortunately ICBN Australia 2011, one of the theme line is hemiparasite, and they have searched worldwide and collect my detail and mail me from IBC, International Botanical Congress, one invitation. You can talk on hemiparasitic taxa. It is a overwhelmed, and you can take with your research scholars, 6 or 7 research scholars. So it's not, it's only my is discuss with my research scholars regarding the Australia Melbourne. So they are interested, but have you but need a lot of money. But they somehow gather the money and move to Australia, Melbourne. We have presented a plenary session with my 5 students. This is a remarkable in my life. Any taxonomist or any botanist, ICBN minded is not only taxonomist, all the botanists. You Arjun, you specialize in cytology, but you can join. I do not know other participants are which domain, but only microbiology, they are separate code, but all the botany have a single code. Now previously it is ICBN, now it is ICN. And why it is our 2011 is remarkable ICBN? Because it's a historical on the point of view. The name has been changed, ICBN to ICN. And all the participants except one lady teacher vote against this resolution. It is also been noted. And a remarkable another point is that 50 participants from China, from postgraduate students, research scholars, and faculty members, they joined in IBC 2011 and voted for China. Our country is a big country, have 4 biodiversity hotspots, have a mega diversity country, have enough populations, but we need good leadership for future, for host one IBC conference, which will be a very much proudful moment for us. We are the botanists. That's all. Thank you.
Arjun, there are several questions in YouTube chat box.
Okay. One question is, what is neotaxonomy?
Systematics or biosystematics, we can call neo-systematics with the help of advanced analytical technique used in taxonomy and solve different types of critical taxonomical problem. We can say neo-taxonomy or neo-systematics.
Okay, sir. Another question, can from participant Subhodip De, can Karnataka and Darjeeling tea distinguish by chemical taxonomy method?
No. Geographical place does not reflect any type of chemotaxonomical work. We can collect plant, analyze, and after that we conclude. Darjeeling, we know that in our experience, in our study, Darjeeling, we know that it is a Himalayan range and have sustainable temperature for particularly molecular and chemotaxonomical work. So you can work easily, and the protein denaturation is not possible, but in plains, protein denaturation is always in common happening. So with the help of 4-degree temperature, you know that, Arjun, Mumtaz, yes. I have established one laboratory, artificial laboratory, that is 4-degree chamber.
Yes, sir.
Particularly molecular systematics and chemotaxonomy. So if any candidate or any participant can move towards systematics with the help of chemotaxonomy or molecular systematics, have good, adequate laboratory is required. Without instrument, sophisticated, it's not possible. And geographical location is not significant in case of our study. We can collect the sample and used in our objective.
Another question.
Please.
How Sementides serological study is different from mammalian serological study?
Sementides
serological study is different from mammalian serological study.
My topic, my observation, my research only on plant. You can ask any questions from plant. from my research, my experience, I will try my level best. But if we put in a mammal system, it's not possible to answer. Science is not hypothetical. It is practical. Please.
Okay, another question. If environmental stress affect the protein content, then what will we do in the research or in chemical taxonomy?
Pardon?
If environmental stress affect the protein content,
If environmental
stress,
yeah, environmental stress
affect the protein content,
yes,
then what will we do in the chemotaxonomy?
yes, very interesting question and very intelligent question. Mind it. When any type of environmental condition will affect protein, not only DNA, not DNA. So we have to collect plant sample and homogenized in a same buffer system, okay, and use it. Any type of environmental changes does not affect in our gel system, because gel system is totally a buffer system and have specific pH. In case of, we know that two types of buffer system, one native gel, another is STS. In case of DNA binding patterns, we are commonly use sodium dodecyl sulfate, because all the proteins are in same nature. Previously, we know that protein have either negative or positive. But in case of sodium dodecyl sulfate, it is a molecular detergent. All the protein bands are in the same direction. So that's why, and we know that, and in case of native gel, with the help of 4-degree temperature, it's not used in chemotaxonomical work. Only protein nature we can study with the help of page or native gel. So environment, that's why we create a artificial environment 4-degree for protein and enzyme. Any questions? Hello? Hello?
Participants can directly interact with our
yes,
sir,
no
Professor
problem.
Amal Kumar Mondal. Participant, please, if you have any question related to subject or topic, you can interact with the
Yeah, this is the great
research
opportunity.
person.
It's not possible,
Yeah.
tomorrow.
Sir, dear coordinator, please. Can you hear me?
Yes, hear me.
Sir, please share your PPT in WhatsApp group, please.
It's not possible from my side. On the organizers, they have my lecture already recorded and put in YouTube. You can collect or connect YouTube for download. Okay.
Okay, sir. Okay, sir. Thank you, sir.
You are from which place?
Sir, I'm calling from Tamil Nadu, sir.
All are from Tamil Nadu.
So, sir, I'm Nitya. Again, I'm Nitya.
Okay. You are from BSc or MSc?
Sir, I am assistant professor in Sita Lakshmi Ramasamy.
You are the faculty members.
Yes, sir.
So
yes,
you just take classes in your class for chemotaxonomy because it is interesting.
Yes, sir. Yes, sir. Sure, sir. But I am not aware of this lecture, so that's why I'm with you for past 2 hours. Sir, you shared so many things. Really admiring lecture. There is no words at all.
Thank you, ma'am.
Thank you, sir. Thank you so much.
One best friend from Tamil Nadu, Professor Ramachandran from Coimbatore University. He's my best friend.
Okay, sir. I heard about him, but I don't know.
He's a nice person.
Yes, sir. Yes, sir. Are you talking about HOD, sir?
Yes, yes.
Yes, sir. I know him. I saw him, but I don't know. We are not aware of each other. That.
But you should develop yourself if you are interested about this line.
Sure,
So
sir. Sure, sir.
And very interesting.
I'm working in you. I'm working in. I have in my college. We have only UG department, sir. So there is no way to research work. So that's the problem. But
we are the academicians.
We are faculty members are interested to doing research.
Yes,
we are applying so many projects and doing some research work.
You have completed your PhD?
Sir?
Do you have completed your PhD?
Yes, sir.
From which topic?
Actually, I did everything in tissue culture, antimicrobial studies, and antioxidant studies, sir.
Oh, very good. And based on antioxidant percentage, you can also classify plant.
Sure, sir. Sure, sir.
Yes. Very interesting.
I will do in future, sir.
Yes, yes, yes.
Thank you so much for sharing, sir.
I will help you. No problem.
Sir, please share your address, mailing address, and phone number, sir.
Okay, okay, sir.
In chat box. Thank you, sir. Thank you so much.
Okay. Thank you. Welcome.
I request participant, if any question in your mind, please ask sir directly. If any query related taxonomy, not only chemotaxonomy, taxonomic field, any question, please directly ask sir. If any question from participants. I have one announcement. Subhadeep De suggests lucid lecture share. Thank you. Please arrange a workshop in future on medicinal plant of South Bengal for UG or PG students.
Very good suggestion, but the practical feasibility, I will try my level best, but I do not know. So, but thank you for your suggestion. I have the mind. I have this with medicinal plants of Southwest Bengal. So it's not possible 1 or 2 days. It's 1 week is required. 2 days for field and rest of the days for practical.
If any question, request participants, if any question or query about taxonomically field, so you can directly ask the question, Mondal sir. Sir, from Dr. Jyoti sir, what is the relevance or application of phylogenetic studies? or what software you are using? From YouTube chat box.
There are so many software in the free version in the website. and its duration is just 1 day or 2 day or 1 week. It's not possible in your PhD. I do not know do you have PhD or not, or your research scholar. But good software, FileIt, you can purchase. But in our research, when I was in research scholar, or my postdoc also, it's not possible to purchase, or anybody from taxonomy or those who are commonly used software, they did not help any type of on the software point of view. So I have developed myself in my laboratory, myself also, for analysis. So you can collect file from purchase or any type of software are in in a website. Few are free till now. Okay. And your previous question, please. Pardon. Utility of phylogenetic.
Okay, sir.
Utility of phylogenetic. Without phylogenetic study, we can't say nothing. Previously, suppose you are the botany student. One example. Previously, we know that woody angiosperm is the origin of angiosperm, like magnolia, trichodendron, tetracentron. All are maximally present in northeast, this area. And when Professor, when the Taktajan visited this place, he categorically told this area is called cradle of angiosperm. Maximum number of primitive angiosperm, particular this area. But now, based on molecular taxonomy, with the help of analytical analysis, phylogenetics based, now it is, the concept is 180-degree changed to herbaceous origin. So that is the utility of phylogenetic. Without phylogeny, it's not possible to conclude any type of decision. So without phylogeny, another thing is that when Linnaeus started his identification, you know that Linnaeus is the father of taxonomy. There is no such type of evolutionary significance or work, only morphological based work. But gradually, in my previous introductory lecture, I have told categorically, gradually taxonomy has developed enormous angle on the side by side with the help of molecular techniques. And now that's why APG4 is possible, APG1 is possible, APG2 is possible, APG3 is possible. Such a long, short gap, 4 classification based on molecular data is available in the net, is available in the world, and accept not only in world, IBC also, APG4, based on phylogenetic system, based on molecular data, particularly cpDNA and molecular DNA, nuclear DNA. So it could not be possible if the technology, if the molecular sophisticated techniques would not be involved with this type of development. So taxonomy is not only the identification of taxonomy, now it is molecular identification. My previous lecture, that is in the Egra College also, the medicinal plants, and in Lady Bourbon College also, medicinal plants identification based on DNA barcoding. So what is DNA barcoding? DNA barcoding, it is the latest technology for molecular identification. So phylogeny is required, basic concept of taxonomy is required, and molecular phylogeny is also required. And you know that the maximum number of questions in NET, National Eligibility Test, for lectureship and for research, maximum questions from phylogenetic chapter, monophyly, polyphyly, paraphyly. These all the apomorphic, rhizomorphic, these are the terminology, these are the definitions are very much involved with this advancement. When I was in MSc student, only taxonomy and systematics. But now in my syllabus, you can see my syllabus, Vidyasagar University taxonomy syllabus. Enormous data, enormous chapters I have included in my syllabus for my student, NET, SET, or in the all India level examination competition. So taxonomy is now only just identification and submit one herbarium. You know that in all the foreign countries, the herbarium is a traditional herbarium, side by side, barcode with barcode herbarium. You can check any type of developed countries, botanical garden, herbarium. All the herbarium converted to with the help of barcoding and also DNA barcoding. So it is only possible with the help of latest technology, latest molecular biological techniques. Our faculty members throughout India, throughout the world, you just search website, Google or any type of search engine, tremendous development in plant systematics. But in our state, it's not that enough development on the taxonomical level. Throughout India, only you can find out 5 to 10 taxonomists, those who have also the molecular background, chemotaxonomical background. It is also required. And our next generation, we have to train our student, our research scholar for future, because future is the plant systematics. You can use chromosome data for any type of disputation. You can use any type of palynological data. You can use any type of new antioxidant used with the help of numerical taxonomy used in our laboratory. Is a surprising result from the nanotaxonomy. Our one team from MSc student, they are now fully established in different school colleges. They first detected the nanotechnology, silver nanoparticles, gold nanoparticles. With the help of numerical taxonomy, we construct a new classification. One of my research scholar, now is in university also, that is uh, the can't remember the university that is in Salt Lake. So he worked on that is calcium oxalate crystals. You know that it is under microscopy so tiny. It's not possible visible in a open eye. So with the help of ultrastructural study, same time, we analyze entire Araceae group family and classified in a different angle, and world is accepted this classification Araceae. So it is a new approach. You should think fast and applied with the help of techniques, with the help of molecular applications, and find different types of tremendous new discoveries. We are always searching this type of plant related attributes for future application. You are the just starter of the assistant professor. Now you are associate professor and professor. So you have enough future to develop this type of new branch of science. I can help, no problem. You just established, you think fast, you can enough excursion, field work is required. Without field, it could not be possible to identify plant. Your think is related with the problems of plants, which plants is similar or dissimilar with the particular family. One thing is that Professor Conquest splitting his family, his classification in tremendous way. Just for example, Cascutaceae family, you know that Cascuta belonging to Ipomia, that is convolvulaceae. But only single character, pollen character, he shifted to this plant to a Cascutaceae. And ICBN has accepted his contribution. So you can think this, any type of taxonomical problem, any type of morphological problem, any type of other problems, you can solve with the help of chemotaxonomy, with the help of molecular taxonomy, or with the help of additional SEM or TEM. These two instruments is so costly, it is not possible to purchase each and every institute. But we can collect, or we can use our sources for our data. Because we are the botanists, we can use in different angles. We have different types of specialization, but our thing is so unique. So we can use all the characters for solve this problem. That's why it is called taxonomy is a synthetic subject, integrated subject, and Conquest Thorn Dahlgren called integrated system of classification with the help of different types of plant characters. Any questions? Hello. Hello.
If any question and query from participants.
মনে হচ্ছে আর কারোর কোশ্চেন নেই। I think no question at all.
আমি একটু জল খাচ্ছি, গলা শুকিয়ে গেছে।
অর্জুন, হ্যালো।
হ্যালো।
আর মনে হয় কেউ নেই, এবার ফার্দার এগাও।
হ্যাঁ, এবারে এন্ডের দিকে গেলে ভালো।
হ্যালো,
হ্যালো।
আর কোনো কোশ্চেন থাকলে বলো।
Hello, Momtaj ma'am, are you audible?
Yes. Conclude the session.
Yeah.
I think there is no question, so I conclude the session. At first, my grateful thanks to our chair, Professor Dr. Amal Kumar Mondal. We know there is a vast subject of taxonomy and there are many part and branching of taxonomy. I request sir, please focus on modern trend. Sir also focused on chemotaxonomy, and today it is a very relevant topic to our student, research scholar. Many research scholar work on that field and complete their PhD also under our sir guidance on that topic, and many research scholars continue their research in that field. So sir try this session very easily and very informatively with their presentation slide is more effective to our participants and our research scholar academician. I think there is a most informative lecture and lucid presentation. So I hope this is very helpful in present and future CBCS or postgraduate, undergraduate syllabus. And everyone can see and observe this lecture in our YouTube session when you necessary in that related topic. So again, thanks, sir, with sharing your views and expending your valuable time in our 1 week lecture session. Before ending this session, I request sir for a song because he engaged in this culture from many days, and our participant also request to you. So sir, please one song from you.
Thanks, Momtaj, from me and Arjun.
Okay.
কখনো তোমরা বলছো যে ইংরেজিতে বলতে হবে, এখন কিসে গাইতে হবে সেটা বলো।
না স্যার, বাংলাতে রবীন্দ্রসঙ্গীত।
রবীন্দ্রসঙ্গীত?
ইয়েস স্যার। এটা সবাই বুঝবে স্যার, সুরটা সবাই বুঝবে।
আকাশ ভরা সূর্য তারা, বিশ্ব ভরা প্রাণ, তাহারি মাঝখানে আমি পেয়েছি মোর স্থান বিস্ময়ে। তাই জাগে জাগে আমার গান আকাশ ভরা সূর্য তারা বিশ্ব ভরা প্রাণ তাহারি মাঝখানে আমি পেয়েছি আমি পেয়েছি মোর স্থান বিস্ময়ে তাই জাগে জাগে আমার প্রাণ আকাশ ভরা সূর্য তারা বিশ্ব ভরা প্রাণ তাহারি মাঝখানে আমি পেয়েছি আমি পেয়েছি মোর স্থান বিস্ময়ে তাই জাগে জাগে আমার প্রাণ আকাশ ভরা জানার মাঝে অজানা রে করেছি সন্ধ্যা বিস্ময়ে তাই জাগে জাগে আমার প্রাণ আকাশ ভরা সূর্য তারা বিশ্ব ভরা প্রাণ তাহারি মাঝখানে আমি পেয়েছি মোর স্থান বিস্ময়ে তাই জাগে জাগে আমার গান আকাশ ভরা।
Excellent, sir. Excellent.
Thank you, sir. Very excellent.
থ্যাংক ইউ। তোমরা আমাকে দিয়ে লেকচার দেখাচ্ছ, গান গাচ্ছ।
না স্যার, এটা আপনার মানে এক্সট্রা,
সেটা আবদার স্যার, আবদার স্যার।
এটা সবার মধ্যে নেই স্যার, অবাক হয়ে যাই আপনার এরকম গলা।
না
হ্যাঁ,
ভালো লাগলো তোমাদের সঙ্গে ইন্টারঅ্যাক্টিভ সেশন, এবং এত কঠিন চ্যাপ্টারটা কতটা ছেলে মেয়েদের যা কোশ্চেন দেখলাম তাতে খুব ইন্টারেস্ট পেয়েছে, এইটুকু মনে হলো। দেখা যাক।
Sir also get their degree from Vishwabharati University. So thank you, sir, for your nice song without any instrument, only of empty voice.
yes.
So we end our session because sir joined with more or mostly 2.5 hours. So again, thanks, sir, and
থ্যাংক ইউ স্যার।
থ্যাংক ইউ ভালো থেকো সবাই, আবার দেখা হবে। তাড়াতাড়ি সিচুয়েশন থেকে যেন মুক্তি হয়, কোভিড থেকে। ঈশ্বর আল্লাহ ভগবানের কাছে প্রার্থনা।
Yes, sir.
ভালো থাকবেন
সবাই সুস্থ থেকো, ভালো থেকো।
One announcement, we again all meet in our 7th lecture session tomorrow 10 a.m. from same Zoom link. So participant also attend in tomorrow lecture session. Thank you for participant who join from 10:30 a.m. to 12, already 12:30 cross. So thanks to all again. Thank you.
Sir, please share your details, sir, in chat box.
এই আমার ডিটেলটা মুমতাজ দিয়ে দিও।
Okay, ma'am. We also provide in our WhatsApp group.
Thank you, ma'am. Thank you so much.
Yes.